Okay. Hi, everybody. This is our final project. We decided to work on Ebola outbreaks. My name is Gabby.
I'm Jasmine.
And my name is Liba.
We are in Virology, MCB 4503, and we are group number 1. So, what is Ebola virus? Ebola virus, along with the Marburg viruses, is part of the family Filoviridae. The word philo means filament, and this family is named as such because of the filamentous shape of the virion. The filoviruses are part of the order Mononegavirales, which make up the negative sense RNA viruses, but we'll get to that afterwards. On the right side, we have a graph that shows the 5 different serotypes that are infectious to human beings. Over here, the first virus is the Zaire Ebola virus, which is the most fatal and the most common. It is also the cause of most of the outbreaks, and it has a 68% mortality rate. The second virus, the Sudan Ebola virus, has a 53% mortality rate. Both of these kill over half of the people that they infect. The Mundi Bugyo Ebola virus kills about a quarter of the people that it infects. And lastly, the Reston and Thai forest Ebola viruses kill 0% of the people that they infect. Each strain of the Ebola virus comes from the place that it was first identified. The Ebola virus name itself is named after the Ebola River of the Democratic Republic of the Congo, near where it was discovered. The Sudan Ebola virus is named after the country it was first discovered in. The Bundibugyo strain comes from the name of the district in Uganda where it was discovered. The Reston Ebola virus was discovered in Reston, Virginia, in primate facilities, and the Thai Ebola virus was discovered in the Thai forest of the Ivory Coast. Ebola viruses have rather non-specific symptoms, especially in the beginning. These include fever, fatigue, muscle pain, headache, and a sore throat. More severe symptoms of Ebola virus include diarrhea, vomiting, rashes, and internal or external bleeding, according to the World Health Organization. This is a very painful virus to have, and the average mortality rate of the virus is about 50%. But during outbreaks, the mortality rate can be anywhere between 20 to 90%. A large part of such a wide range may be due to the type of Ebola virus that causes the outbreak, since not all serotypes are equal in fatality, as we just saw on the last slide. Transmission of the virus is through direct human contact, but can also come from infected animals. On the right, we have a picture of fruit bats, which may serve as a vector to spread the disease across Africa, the main continent where Ebola is found. One interesting thing about the virus is that as long as it is present in the blood, it is infectious. This means that it can remain infectious long after an individual recovers from the virus, and that it can be transmitted after death. There have been reports of healthcare workers working closely to an infected person, as well as people involved in taking care of the dead body and preparing it for burial that have been contaminated. Unfortunately, it's also possible for the virus to be passed on vertically or from the mother to the offspring. If a woman that has or had Ebola gets pregnant, there's a high chance that the child will be born carrying the disease or even with the disease. The first recorded cases of Ebola happened in Africa in 1976. The first outbreak was in the village of Yambuku in the Democratic Republic of the Congo in Africa. The species was the Zaire Ebola virus with an 88% mortality rate. There were 318 reported cases, and transmission was mainly contaminated needles and syringes in the hospital, as well as close personal contact. The second Ebola outbreak was in Sudan with 284 reported cases, and it is believed that the outbreak began in a cotton factory and then spread through close personal contact in hospitals, affecting many healthcare workers. So, the genome and the virion structure of the Ebola virus. The virion of the Ebola virus in general is filamentous, hence the name filoviruses. The Ebola virus virions are enveloped, and the genome is about 19,000 base pairs long. The genome is linear, non-segmented, negative sense RNA and encodes for 7 different structural proteins. Starting here at the 3' end, we have the nuclear protein, which, along with viral proteins 24, 30, and 35, and the RNA-dependent RNA polymerase create the ribonucleoprotein complex. BP24 and the polymerase, however, are closer to the 5' end of the genome. Viral protein 24 is associated with the membrane of the virion and messes with the signaling of interferons. Viral protein 35 is an interferon antagonist, like another line of defense against interferon after BP24, and is found in the polymerase matrix. The RNA-dependent RNA polymerase aids in the synthesis of new RNA strands from the RNA template. The little yellow spikes that we see here are on the virion, are the glycoproteins that help the Ebola virus attach to the cell and enter it. Another interesting thing about Ebola viruses is that in addition to the glycoproteins on the surface, they also produce soluble glycoproteins that are secreted from infected cells. These soluble glycoproteins set Ebola viruses aside from other mononegaviruses.
Okay, so moving on how the Ebola virus actually enters the body, there are three main ways it gets in. First, through the mucous membranes. This is basically soft linings inside the areas like the eyes, the nose, and the mouth. Second, it can enter through breaks in the skin, even like small cuts we might not notice. And third, through direct contact with infected bodily fluids. This includes things like the blood, feces, or vomit from someone who's sick, which is why Ebola spreads so easily in healthcare settings and during funeral practices where people aren't wearing the proper protective equipment. Now, once the virus is inside the body, its first targets are immune cells near the entry site, specifically monocytes, macrophages, and dendritic cells. These are all types of white blood cells that normally help detect and fight infections. However, in this case, Ebola actually hijacks them to spread itself further into the body, which kickstarts inflammation. On the surface of the virus is a protein called glycoprotein. It binds to specific receptors on the surface of host cells. The virus is then taken in mainly through a process called macropinocytosis, which is a type of endocytosis where the cell kind of like drinks in large amounts of fluid along with whatever's in it, for example, the virus. It can also use receptor-mediated endocytosis, where it enters by binding to a very specific receptor that triggers internalization. Both of these pathways actually rely on the PI3K AKT pathway, which is a signaling route inside cells that control things like cell growth and movement. It also depends on cytoskeletal dynamics, so basically the movement and the structure of cells' internal scaffolding. Then the virus manipulates this to get itself in. And one final point, Ebola doesn't actually infect lymphocytes, which is another major type of immune cell, but it does cause them to die off in large numbers through a process called apoptosis. This ends up weakening the immune cell even more, which makes it harder for our bodies to fight back. So overall, from this, we can see that the virus is actually very strategic. It enters through exposed areas, targets key immune cells, and then uses the body's own cellular machinery to spread. Next slide. So now we know how the virus enters the body. Let's talk about where it goes and what cells it infects. The first line of attack that I mentioned is on the monocytes, the macrophages, and the dendritic cells. These are all types of immune cells that act like the body's early warning system, since they detect invaders and alert the rest of the immune system. But when Ebola infects them, it actually uses them to travel through the body and then spread the virus. This causes a huge release of inflammatory signals, and this contributes to the severe symptoms we see the virus cause. Then the cell takes in the virus through macropinocytosis, which is regulated by the pathway, and then the cytoskeletal proteins help move the virus inside. After that, the Ebola virus goes for the endothelial cells, which lines our blood vessels, and it causes severe damage here because it can lead to vascular leakage. So basically, fluids start leaking out of the blood vessels, and in some worst cases, hemorrhaging or internal bleeding can occur. So next, the virus targets hepatocytes or liver cells. This leads to liver damage, which is a huge problem since the liver helps detoxify the body, and it also makes proteins that help blood clot. And if the liver is no longer working, then the bleeding becomes even worse. It also infects the adrenal cortex cells, which are in the adrenal glands, which sits above our kidneys. These help control hormones that regulate blood pressure and other vital functions. So if these cells are damaged, we see issues like hormonal imbalances, and this can cause organ failure. Next slide. Now let's talk about the actual stages of infection that happens when the Ebola virus enters the body. We start with the entry and the early infection phase. As I mentioned earlier, the virus enters through the mucous membranes or through breaks in the skin. Once inside, it infects our immune cells using mechanisms like macropinocytosis or receptor-mediated endocytosis. But there's also something called lipid raft mediated entry. So this is like lipid rafts, like little patches in the cell membrane, and it helps organize the receptors and make it easier for the virus to get in. Then the virus uses its glycoproteins to bind to the host receptors, for example, beta 1 integrins, which are proteins that help stick to each other and the surrounding tissue. And NPC1, a cholesterol transporter that the virus takes over to get inside the cell. Once the virus is in, we move into the systemic spread stage. The infective immune cell, like the monocytes, travel to the lymph nodes, the liver, and the spleen, and basically carry the virus with them. At this point, the virus has started to replicate a lot, and then we get something called high viremia, which means that there's a high concentration of virus in the blood. The virus also starts evading the immune system, which makes it harder for our bodies to fight back. Then comes the organ damage phase. Ebola starts replicating aggressively in critical organs like the liver, the adrenal glands, and the endothelial cells lining the blood vessels, and this triggers a cytokine storm, which is an overwhelming immune response that actually damages the body. It also causes internal bleeding, and that can lead to multi-organ failure. And lastly, the late stage. In severe cases, patients go into shock, experience widespread hemorrhaging, and if the immune system cannot catch up, it can unfortunately result in death. But if someone survives long enough for the immune system to take control, they can recover. Next slide. Now here we see this diagram where it shows how the Ebola virus induces innate immune cell dysfunctions. In the part A section, the Ebola virus is able to impair type 1 IFN production by infected cells and then block IFN response in the uninfected cells. In part B, the virus is able to induce massive cytokines and chemokines production by the monocytes and the macrophages. And in part C, the infection is able to impair DC maturation and deregulate the cytokine production. And lastly, part D, we see that the virus is able to induce massive NK apoptosis, avoiding the NK function and impairing NK-mediated DC maturation help. Next slide. Okay, so let's get into the molecular side of the virus, how Ebola actually gets into the cell and replicate once it's inside, starting with the receptor binding and entry. So this is the virus's way of attaching to and entering the host cells. Ebola has a glycoprotein, which is a sticky protein on the outside of the virus. The glycoprotein recognizes and binds to specific host cell receptors. The main receptor is TIM1, which stands for T cell immunoglobulin and mucin domain 1. It's found on the surface of many immune cells, and Ebola can also use C-type lectins, which are sugar binding proteins. And once inside the cell, it relies on the receptor called NPC1 in the endosomes, which plays a big role in getting the virus into the right compartment for fusion. Once inside the endosome, the low pH and the host enzymes like cathepsins activate the virus, and these enzymes help cut the viral glycoprotein, triggering fusion of the viral and host membranes. That fusion lets the viral genome escape into the cell's cytoplasm. Now moving on to the replication cycle. Ebola is a negative sense single-stranded RNA virus, which means its genome is like a reversed version of mRNA. So it can't translate directly into proteins. So the virus ends up bringing along a special enzyme called the L protein, which is an RNA-dependent RNA polymerase. That enzyme takes the viral genome and transcribes it into mRNA, which can then be read by the host cell's ribosomes to make viral proteins. And as more viral proteins are produced, the viral makes full-length copies of its RNA genome, which are used to build new virus particles. All of these components then travel to the plasma membrane of the host cell, where they assemble, bud off, and release new viral particles to infect even more cells. Next slide. Okay, so wrapping up the molecular side of things, this slide covers two major topics: the key viral proteins of Ebola and how the immune system responds or fails to respond during infection. So let's start with the viral proteins. Ebola might seem like a simple virus, just RNA and a few proteins. However, each of them play a significant role in helping it survive. spread, and overwhelm the host. So first is GP, the glycoprotein. This is the one we talked about earlier that binds the host receptors and helps the virus fuse with the host membrane to get inside the cell. Then we have NP, which stands for nucleoprotein. It wraps around the viral RNA, kind of like a protective shell, and then helps organize the replication process. VP35 is one of Ebola's biggest weapons against the immune system. It blocks the interferon response, which is a major part of how the cells defend against viruses. It also helps replicate more efficiently. VP24 also helps shut down the immune system. It prevents STAT1, which is a signaling molecule, from entering the nucleus. And without STAT1, the cell cannot properly activate interferon stimulated genes. So again, the antiviral response is blunted. VP30 acts as a transcription activator. It helps start the process of turning the viral genome into mRNA so new proteins can be made. VP40, all about budding, which is how the new virus particles exit the host cell. Without this, the virus cannot spread. And finally, the L protein. This is the virus's RNA polymerase. It copies the viral RNA and it makes the mRNA needed to build all the other proteins. So now let's look at how the immune system reacts. In the innate immune response, the virus initially infects monocytes and macrophages, which are basically the first responders in the immune system. And then this infection triggers a cytokine storm, which is kind of like an overreaction of the immune system, where tons of inflammatory signals are released, and that kind of ends up damaging the tissues instead of helping them. And at the same time, VP35 and VP24 are suppressing the interferon response, which is normally one of our strongest defenses against viruses. So while the body is trying to fight back, the virus is actively sabotaging it. Then we move on to the adaptive immune response, which involves lymphocytes like T cells and B cells. Unfortunately, Ebola causes widespread lymphocytosis, like I've mentioned, or something called cell death. So these critical immune cells die off in huge numbers, which means the body struggles to produce antibodies or mount an effective T cell response. So the result of this is massive immune dysregulation. The body is overwhelmed by inflammation and is unable to control the infection. And this contributes to the severe symptoms like bleeding, organ failure, and in many cases, death. Next slide.
Now we're going to talk about Ebola outbreak history. Thank you, Jasmine. So over here on my right, we have a picture of the continent of Africa. Most of the Ebola outbreaks are concentrated in Africa. So each white country does have a reporting of an outbreak. All of the countries in gray do not. The size of the circles show the number of cases. So the smaller the circle, the fewer cases were reported in that outbreak. Finally, the color does mean something. It means which species we're looking at. So as you can see, most of them are red because most of them are from the Zaire serotype. The blue ones, mainly in the east of Africa, are from the Sudan. The couple of yellow ones in the middle are from the Mundibugyo, and the one from Ivory Coast is from the Thai forest. According to the CDC, there have been 39 outbreaks of Ebola virus in Africa since 1976, including the first two original outbreaks, as well as the West African epidemic between 2014 and 2016 in Guinea, Liberia, and Sierra Leone. There have also been outbreaks in the United Kingdom, Italy, Spain, the Philippines, and the United States. However, many of the outbreaks outside of Africa were due to a patient or the patients working with experimental vaccines and not being careful, or by being infected by primates or other animals that had been infected with the virus. For this presentation, we will just focus on the African outbreaks. The first recorded outbreaks, as was mentioned before, were in Sudan and the Democratic Republic of the Congo. The virus got its name from the outbreak near the Ebola River of the Congo. The Sudan outbreak is the namesake of the Sudan serotype. As was mentioned before, the Congo outbreak was due to the Zaire Ebola virus and had a mortality rate of 88%. The Sudan outbreak was not as severe and only had a mortality rate of 37%. The next notable outbreak was almost 20 years later in 1995, again in the DRC, and again thanks to the Zaire serotype. The mortality rate was a bit lower, 81%. However, the transmission among health workers was curbed significantly when measures against contact were taken, such as gloves, gowns, and masks, just like were taken during the COVID pandemic. In 2001, Uganda had a major outbreak from the Sudan Ebola virus. There was about a 50% mortality rate and was East Africa's first Ebola outbreak. So that's what this is talking. The next outbreak is the largest in the history of Ebola outbreaks. The West African epidemic began in 2014 in Guinea, then spread to Liberia and Sierra Leone. Within 5 months, it was declared a public health emergency of international concern, or PHEIC. There was lots of rapid spread due to poor health measures in this area of the world, and identification of the virus was limited due to the lack of public health infrastructure. Unfortunately, Fortunately, sorry, many worldwide and government organizations such as the World Health Organization and the CDC of the United States, along with many other health agencies, stepped in to help stop the spread and deliver the help that was needed. The outbreak was caused by the Zaire Ebola virus, but only had a 39% mortality rate. In June 2016, the epidemic was considered over. The final main outbreak, and the second largest after the West African epidemic, occurred in the DRC in 2018 with the Zaire Ebola virus. It lasted close to 2 years from August 2018 to June 2020. There were 2,287 reported deaths with a mortality rate of 66%. The virus spread through multiple provinces and even into Uganda. This was the 10th Ebola outbreak the government had confirmed by this point. The DRC government was assisted by our own CDC to help in testing, vaccination, and logistics, and other components that helped put a stop to the break. The final recorded outbreak was in November of 2022 in Uganda, where 164 people were infected with the Sudan Ebola virus. Only about a third of the people who were infected died. So you can see that these major viruses are labeled, in a sense, within each of these. Many of the DRC outbreaks are labeled here. The West African epidemic is labeled here.
Okay, so for current field knowledge, different outbreaks occurred in sub-Saharan Africa, particularly in Central and Western Africa. The largest outbreak that was mentioned by Gabby in the last slide as well, which occurred in 2014 and ended in 2016 in Guinea, Liberia, and Sierra Leone. This was the major and the biggest outbreak. And also here in the picture, we see that the countries are labeled with the dark shaded blue color, and also the light shaded ones. These were the light shaded ones are the ones that were not like that didn't had many cases shown for the Ebola virus, but the dark ones are these three major countries that had this outbreak in 2014 and 2016. For this outbreak, many countries and the organization around the world helped this part of the Africa to overcome this um, uh, Ebola virus. Different organizations such as World Health Organization, Doctors Without Borders, and US, UN, Japan, China, they provided significant medical and financial support to the affected areas. Not only they provided the technical guidance, but they also provided like the coordinated international efforts, and they deployed experts so they can go to the affected area and help the medical and help in medical needs. Also, the United States, they deployed personnel. They also provided the good healthcare system there, so the patients can get the good medical needs. After that, the medical teams from China also went to these affected areas. They provided technical guidance and supported the facility constructions as well. Japan provided the emergency grant aid during that time in to support the Ebola response in Western Africa. African Development Bank, they also combined, I think it was like $220 million that they sent in the support of these countries after this Ebola outbreak. Next slide. So these are the multiple Ebola vaccines that are available, and they are approved by FDA. The first one is Ervebo. It is a live attenuated virus vaccine that protects against the Zaire strain of Ebola. And the next one is Zabdeno vaccine. It is used to deliver Ebola virus genes. This the second vaccine. It has two doses, so it is given one by one. Like first, the patient receives the first dose, and after a few weeks, the second dose is given to the patient. And then it provides protection against Zaire and Sudan strains of Ebola. Both of these vaccines are highly effective in Ebola, preventing Ebola infection and also reducing the mortality rate. The second vaccine that was used for the Ebola virus is also very good in helping is also very good in affecting the Ebola virus, because it has two doses. So with the two doses, people can receive the third dose as well if needed after some time. If they needed the third dose, then it is given to them as well. Both of these vaccines have shown like very highly effective rates in preventing Ebola infection and also reducing the mortality rates. These are generally very tolerated with mild side effects like pain, headache, redness, or swelling at the injection site. Can you go to the next slide, please? Thank you. So for the Ebola virus prevention and control, the leading cause of death in Ebola virus infection is the organ failure. And according to different studies on Ebola patients, the use of acute statin lowers the risk of developing sepsis, which is also known as organ failure. You can, by using the acute statin, we can lower the risk of developing sepsis by 83%, which is a huge percentage. So this is something that can be used to save some lives from Ebola virus. The next effective ways that can prevent Ebola is by washing hands more frequently, uh, also by avoiding contact with the bodily fluid of the infected people. It could be bodily fluid like blood, your urine, saliva, sweat, or vomit. And also avoid touching the dead body of Ebola patients, because it doesn't matter if the person is alive with Ebola or if it's their dead body, you can still get infected with Ebola even if the person is dead. So here in the picture, it is shown that these people, they are handling the dead bodies and they are fully in their protective equipments. They're wearing gloves and masks, so that's how they're handling the dead bodies, and that's how it should be like. So even if you touch it with your hands, you might get infected with Ebola virus. So it is always recommended to use gloves, mask, and all the necessary equipments, protective equipment, so you can stay away from getting infections from Ebola virus as well. When caring for someone with Ebola, always wear appropriate personnel protective equipments. Washing hands frequently with soap is one of the main thing, because if you stop washing your hands during that time when you are handling the patients, you could also get exposed to Ebola virus. So it's always required to use the proper equipment so you can stay safe from getting the infection. Next slide. In most impactful finds, Ebola was identified in 1976. No human cases reported between 1979 to 1994. But as mentioned in the previous slide, the major outbreaks that happened, the first one was happened around like 2001, and then this one was in 2014, and this was the one that was reported in the villages close to like the tropical rainforest in Central and West Africa. DRC, Gabon, and the Republic of the Congo, Sudan, and Uganda, they reported most of these cases. So, and the four species of Ebola virus that causes cases in humans are listed here. The first one is the Ebola virus disease, which is caused by the Ebola Zaire virus from Ebola virus Zaires. This is the first one, the first species, and the next species is the Sudan virus disease, which is caused by the Sudan virus from Orthio Ebola virus Sudanese. And the next one is the Bundibugyo virus disease, which is caused by the Bundibugyo virus Orthio Ebola virus. And the last one is the Thai forest, which is formerly Ivory Coast virus, and it is from the Orthio Ebola virus tie-ins. Next slide.
Okay, so to summarize all of the points, Ebola is a linear, non-segmented filovirus with a negative sense RNA strand. It is part of the family Filovirus and the order Mononegavirales. Ebola viruses, as we saw, are concentrated in Africa, but some cases have been recorded in parts of the world, although it is not due to human contact. Hmm.
And then the next takeaway point is how the Ebola virus rapidly disables the immune system. And this is by infecting the frontline immune cells and then releasing viral proteins that block interferon signaling and trigger massive lymphocyte death. And then the next point we covered was how Ebola's entry and the replication rely on specialized host interactions, like the glycoprotein and how it targets specific receptors like TIM1 and NPC1. And then the entry mainly occurs via macropinocytosis. And once inside, it hijacks the host cell machinery to replicate and assemble new viruses, especially in vital organs like the liver and the adrenal glands.
The next point is the two FDA-approved vaccines that are available, and they are approved by FDA. It's the for Zaire and Sudan strains of Ebola. And effective ways to prevent Ebola infection is by washing hands, avoiding contact with the Ebola patients or their bodily fluids.
And these are our references. Thank you very much, and we hope you have a good day.